In this lesson, we'll continue with some evidence supporting the reactivation hypothesis. We said that retrieving a memory requires reactivating the neurons involved in storing the memory. And in this lesson, we'll look at the case of imagery. We'll pay special attention to visual imagery here. Our capacity for imagery can be explained by the reactivation hypothesis. First, some background. We've learned that the visual cortex then is activated by inputs from the eyes. And of course, the cortex, one of its main jobs in the visual cortex is to identify objects, right, that we see in our world. Of course, this happens with our eyes open. And if we see something we've seen before, we call that an act of recognition. The current pattern of visual cortex neurons somehow matches a stored memory for an object. And we say, yes, I recognize that. I've seen that before. For the purposes of this lesson, we need to quickly review the working memory system. Whatever's in this box is sort of what we're currently conscious of. This is this sort of memory buffer that we have. And the way that we're going to understand this is that other parts of the brain can set up loops of neural activity. Other parts of the brain provide the inputs to the working memory system. So, for example, in a simple case of looking at a tree, visual information will be sent to visual cortex, and if I've seen the tree before, then I will recognize this thing. Yes, that's a tree. Tree neurons will be activated. They will send electrical signals down long axons to frontal cortex neurons, the working memory system. Those neurons in the frontal cortex will respond and send activity back to the visual system, so we'll get these loops of activity. And this is what it is then to be conscious of our tree. The working memory system and consciousness are closely associated. So long as we're looking at that tree, we're going to have a conscious perception of the tree. Now, if we shift our gaze to the cat down here, well, then the tree neurons will no longer be activated. So we're going to stop the tree loops and we'll activate the cat neurons and then engage loops of activity with the working memory system. So our conscious perception will change. We'll be conscious of the cat and no longer conscious of the tree. And it's really striking to see examples of just how dynamic the brain is. So I'll show you this here in a moment. Now, we have seen this video before. We have an immobilized fish here. It's alive and aware of its surroundings. It's tracking this single-celled paramecium. That's the prey. Notice the eyes are changing their position. And over here on the left, we see real-time activity of brain cells. So the cells have been engineered to sort of flash a little green light when they make electrical signals. But you get this sense that, of course, that the pattern of neural activity is quickly changing just from the simple act of changing the direction of gaze of the eyes. Okay, so what about imagery then? What's going on when we imagine something? After all, we can close our eyes and yet sort of see things in our mind's eye. Are we engaging the visual system as well when we make a visual image of something? If so, then imagery is a case of memory retrieval. So the question would be, is the visual system activated when we imagine things? If so, then the areas used to perceive objects, in this case visual cortex, are also involved in making visual memories. And that then would support the reactivation hypothesis. On our sort of cartoon brain, the idea here would be is if somebody asks us to imagine your cat, well, that cue in the working memory system can activate your long-term memory system. So we've got a cat memory trace here. When the cat neurons are reactivated and they set up the loops with the working memory system, now we are imagining a cat. So this would be an example of retrieving a memory. So imagery then, on this reading of the situation, imagery is an example of retrieval of a memory. It's just that we're not using our sensory equipment to recognize something. We are reactivating a memory trace and experiencing it in our mind's eye. So the reactivation hypothesis makes a prediction about imagery. Brain areas used in perception, like the visual cortex, are also used to store memories, visual memories, which get reactivated when we imagine things, a form of memory retrieval. So what's the evidence for this reactivation hypothesis here in connection with imagery? The first line of evidence will be stimulation evidence. We'll go back to the early work of Wilder Penfield in the mid-1900s. He was a neurosurgeon. Here we see the exposed human brain. The patient was awake and conscious. And Penfield, these are some of the first studies of this kind, he would stimulate the surface of the cerebral cortex and simply ask the patient what happened. And what apparently happened was the subject, depending on where the stimulation occurred, would have what sort of were like retrieved memories, experiences that were similar to retrieved memories. In fact, he noticed a pattern. When he stimulated areas near the auditory cortex, the retrieved memories tended to be auditory in nature. When he stimulated visual areas of the brain, the retrieved memories tended to be visual. Now, they might not be memories from the person's life, but they had characteristics like it was a memory being retrieved. Notice the pattern, though, of results supports the reactivation hypothesis. The reason that stimulating the visual areas produced visual memories is because the visual memories were stored in the visual areas, and likewise with the auditory memories. The second line of evidence would be the evidence from damage. Here's the underside of the human brain. There are a couple of regions outlined here where damage produces certain symptoms. So damage to this area can produce cortical color blindness. Subjects no longer sort of see the world in color. Object agnosia would be they have difficulty recognizing objects, and prosopagnosia would be face blindness, difficulty recognizing faces. The interesting thing here is that there is an association. That damage to these areas not only produces difficulty in perceiving the world, but also imagining that quality. So, for example, damage to the color area here means that the subjects no longer perceive the world in color, but neither can they imagine color. For faces, subjects can't recognize faces, but neither can they imagine faces. So this connection between perception and imagery supports the reactivation hypothesis. The reason they can't imagine a face is because the memory traces for faces has been destroyed. So they can't recognize faces, neither can they imagine faces. Let's stay with the face perception here for a moment. Here we have this woman is looking at this guy's face here. We have the visual cortex being engaged, and the visual pathway runs down through the temporal cortex. Here's the fusiform face area, FFA here. So this is this face area that we've been talking about. In our cartoon brain, the idea is when this face area is activated by looking at a face, there's Joe, the neurons here in this part of the visual cortex send signals up to the working memory system, and we get those loops of activity, and we're consciously aware of seeing Joe's face. So this would be perception. The question would be, what happens if we close our eyes and imagine Joe's face? So when we're closing our eyes, right, we're stopping the visual flow of information. But when we imagine Joe's face, the idea here is we're going to reactivate a memory trace for Joe's face and then send that up to the working memory system. The question is the face area activated when we imagine a face? To find that out, we'll introduce another kind of recording technology, the functional MRI scanning. And this is a brain scanning technology that involves putting your head in this giant magnet, and the machine has the capacity to detect changes in brain activity in real time. Now, it's a bit of an indirect measure of brain activity, and just briefly here we have two neurons next to a blood vessel. When neurons become activated, when they're working hard, doing electrical signals and things, it turns out that the local blood vessel increases in size so as to bring more glucose and oxygen to feed the neurons. So the fMRI machine is actually detecting changes in blood flow, but that's an indirect measure of changes in brain activity. When brain cells are active in one part of the brain, they recruit a larger blood supply, and that's what the fMRI is detecting. So in the early research on the face area, here is a researcher scanning her own brain on different days. And again, while in the machine, she would be looking at a computer screen, and they would either put a face on there or some other object, and then a face and an object, face and an object. And this kind of record here, this peak, indicates changes in blood flow. So when a face was on the screen, the face area showed increased activity, increased blood flow to that area. When they put an object on the computer screen, reduced activity. Put a face on, increased activity, and so on. So this was some of the early fMRI data on identifying this face area in the brain. So the question then is going to be, what happens when the subject is not looking at a face, but when they're closing their eyes and imagining a face? Will the face area be activated? So let's take a look. Here we see the comparison between perception and imagery. And we do see face area activation, although not as much as when you're actually looking at a face. Nevertheless, the face area is responding when the person is imagining a face. So we do get activation of the face area. That supports the reactivation hypothesis. Now, why don't we get as much activity? Well, the reduced activity in the face area makes sense because the area is not receiving inputs from the visual system, right? So the face area oftentimes is getting lots of activity from earlier visual areas. But when we're imagining a face, that pathway is not currently active. We are internally activating the face area here, and so it makes sense that there's less activity when we imagine a face. In fact, this may also help to explain some of the variants that we see. Some people have very vivid imaginations, and others it's less so. Some folks say that when they're imagining a face, it's kind of dark in there, and others say that they can see vivid detail of the things they're imagining, and that may be related to the amount of activity in the face area when we're imagining, but more work has to be done on that. So in conclusion then, imagery does support the reactivation hypothesis. Those regions used to process stimuli are also used to reconstruct memories that involve those stimuli, perhaps storing memories. So the face area is activated when we're perceiving a face, but it's also activated when we're imagining a face. And if we think of imagery as memory retrieval, that suggests that these areas of the brain are also the sites of storing memory traces for that kind of stimulus.